Thank you to Perkins for this wonderful conference. Thank you mostly to the audience members, either those affected with CVI or with children or caregivers that have children. It is a journey for me, which you'll see here, some of it, getting to this point. And I hope to convince you that there's real reason for hope, that we can define the things that we all experience in these children in a more quantitative way, and I hope that'll build a foundation for us to truly understand what this phenomenon is and to treat it head on. So thank you very much. So as you can see from the title, the talk today is going to be about quantification. So you're going to hear this word a lot. This is what most people think of when they think of measuring vision. We've all had this. This is an acuity task. It was invented in 1862. It is the most widely used task in all of vision. And there's a good reason for that, because it was designed originally to test for the ability to refract our vision. So our visual systems are a combination of an optical device and a neural device. We see with our brain, but the light in the world out there has to pass through a series of lenses and project on our retinas. And what we do with this acuity task is we read down the chart until we find the place where we blur the actual letters together. And then that's the point at which we go, yeah, you can't see, and we try to correct the optics for that. Acuity, though, has a number of downsides. One is it has extreme limitations for quantifying the vision that has to do with brain vision. It's great for refraction, not so great for this. In fact, many visual diseases, acuity is not a very good and sensitive measure of that. In addition, it's not a comprehensive measure of what I call spatial vision, the ability to see form. You see me up here because I am approximated on your retina as light, but then quickly our visual systems start to parse things into space. The size, the contrast of things is a first-order approximation of seeing form and pattern in the world. It's not a comprehensive measure. It does one aspect of that, but doesn't do it comprehensively. It also has the same problem at the level of treatments. Many treatments we have used for every clinical study on remedying vision in one way or another uses acuity, but it turns out it's not that great a predictor of outcome because of limitations in its ability to capture how the brain forms an image of us as a pattern. And a real big problem, I put it in red, that affects everyone here, one way or another, is that subjects have great difficulty in this task unless you know language. I can't know what you see. I can only infer what you see by your actions. And the actions here are speaking or pointing to, but in some way there's the cognitive ability with language. Now, there are a variety of off-label versions of this, but they're still imperfect because they require that ability to understand and act. And I think I'm seeing a lot of heads nodding here, and I think this is what I'm going to go and tackle head on. So what we have here is many neural diseases aren't well defined. Does that ring with everybody here? And it's not just the disease we're here to talk about. It's not possible to place everybody on the same scale. We'd like to know. When I say to you, well, my acuity is this and your acuity is that, we can put you on the same scale. If we can't measure it or measure it in a different way, how do we know we're talking about the same thing? So it's important that I think it's important that we put everyone on the same scale. As I alluded to earlier, we lack the ability to evaluate the efficacy of promising treatments. If we can't measure whether a treatment is, say, 10%, 20% effective because there's too much noise, how do we know we're even on the right track? So I think quantification is a double-edged sword, or two sides of a coin. We can't measure the thing we want accurately, and we can't measure very well whether we can treat that effectively and get an answer. So I think we need a better way of measuring routine visual function, and I'm going to tackle today two aspects of that. One is spatial vision, this ability to see pattern and form in the world, and the analysis of gaze. That's the thing that we have to use to infer ability to see in one way or another. I'm going to talk about that. So, it turns out that current evaluations in non-communicative subjects have to use this portal, this ability to evaluate gaze in one way or the other. And this is how it's done mostly, with a neurological or neuro-ophthalmological exam. This is a redacted actual neurological exam or neuro-ophthalmological exam. And I actually ask you a couple of things. First of all, it's a photocopied piece of paper. Next, on the right-hand side, you can see that there's a code for a variety of things. And you read those things, and they're words with acronyms. We talked about acronyms earlier in here. And they're meaningful. I'm not saying they're not. But look at the lack of numbers. It's not like, well, how much of this or how much of that. Then when you read the actual exam here, you can see that there's some words describing phenomena. There's a few numbers here that have to do with, placement of eyes and strabismus and so forth. But you get down to this, and at the end, then, finally, you see this. There's the word, or the acronym right there, CVI. This is typically how CVI is diagnosed in a non-quantitative way using this sheet. Now, I'm not, in no way am I saying that there are well-meaning people that are trying to do this, and this child may indeed have CVI defined in other ways. But you can see the problem. We rely on non-quantitative means to do this. And so how could we know if we come back a year later whether there's been some change in this sort of thing? We think we can do better. So the premise we're working under here is where there's a need to objectively, not subjectively, objectively quantify visual function in anyone, but certainly in brain-injured children. And to characterize how those change over time. We're talking about children here often, and children grow, children develop. How do you know if a change over time is a result of their development or the result of a change in the disease state? We can only separate those two if we can measure them quantitatively and evaluate them separately. And we need a way of better defining, diagnosing if it's present, grading it so that we can put it on a scale before we can, I think, really effectively evaluate treatments. So I'm seeing lots of heads nodding in the audience here. I hope I'm preaching to the choir. So why am I here? I'm not here because I have a long history in clinical work in CVI. It's a rather short history. I am a biology geek. My background is in basic neuroscience. And when we say basic neuroscience, we mean pretty much rodents. I've been working on rats and mice for decades. This is what we do. We try to understand in those models of how brain works at all, in my case, the visual system. We mimic diseases in a variety of different ways, often genetically, and then evaluate that, and then use that information to try to apply, we call it translation, to the human condition. What I'm and my lab is known for is having developed ways to assess vision in rodents. Like I tell people, it's not the sort of thing that you really go into with the person sitting beside you in the plane, right, when you're waiting there on the tarmac or something. But it turns out that people do this, and we're quite proud of it, and we think it's an important thing. So, remember I said I can't know what you see, I can only infer by your actions? Well, one way that we can infer visual function is through, we call it smooth tracking, the ability to follow information smoothly in the world. You can't fake that. You need a target on the retina. You need a stimulus out there in the world that moves across the retina, and then you can move your eye smoothly. You cannot do that without that target on your retina. So it turns out that if you have an object in the world and you smoothly track it, then gives you high quality evidence that you saw that object and not something else. So this is pretty interesting. Also, it removes a lot of the machinery. There's still a lot of neural machinery between the stimulus and your response. It just doesn't rely as much on the higher order cognitive, cortical sorts of things. So it's got strengths and limitations, and its big strength is it doesn't require overt communication. I talk to my rats and mice a lot, and they don't say much back to me. So if we're able to do that in in these rodents, I think we're all mammals, and I think it can be applied more broadly. This is a mock-up of a mammalian sort of system of why it works. Out there in the world, you have information, you got a couple of eyes, talk to the brain. That information is inverted through the optics we talked about onto the back of your eye, onto your retina. And so when you move that information in the world, and if, let's say, you didn't move your eyes, if you didn't move your head, neck, or you didn't move the eyes at all, what would happen? Well, what would happen is that information is blurring as it goes across, and this is the sort of thing you'd see. However, if you move smoothly with that information that's there, it turns out you see it clearly because you move your eyes and it remains relatively stable on the back of the retina. This is a wonderful invention of nature that allows us to see information by placing it on the retina, but moving that retina as the information moves. Because if it isn't stable on the retina, you don't see it all. We are known for having developed what we call a virtual optokinetic system. It's basically we place rats or mice into a virtual environment, and it's a computer screen walled room that we put them in on a little platform that freely can move, have a camera from above. What we do is this. We take these animals and create the illusion using gaming technology that they're in a virtual world. They look out and it looks like they're in a cylinder. And if you're a vision scientist, you must put gratings on everything. It's just what we do. This is called a sinusoidal modulated grating. Stripes that are black and white, okay? And so from the animal's point of view, there's this cylindrical world that's out there in the world. And what we do is, in the image animal above, let's say you're getting your acuity measured, and you're looking at that chart. And they go, okay, C's and G's, yeah, it doesn't look quite good. You move closer, of course, that information is larger in retina. You say, yeah, that's a C. Well, we have to control the distance. The way we control the distance in these animals is, if the animal's over here and it's closer to these screens over here than it is these, we move the world, literally, for them. Then if the animal moves over here, we move the world. We move it here, we move the world. So we can always know what the distance is. It's in virtual space, but the size of information on the retina. And then what we do is project information on the screen, those gratings, and we move them. And when we move them, if we see the smooth tracking occurs, that's high quality evidence the animal saw that. And then we play the same game as you do in an acuity chart. We make it harder to see. We make the information smaller, we move it faster, we change the orientation, we make the contrast different to get these quantitative measures of these functions. It's very simple, but it turns out it's very effective. So this approach has been adopted worldwide in neuroscience as pretty much the way that we now are able to quantify visual function in rodents. It's become a standard in the field. I've had a career in this for decades. Everything was going fine. Then something happened. Something remarkable happened, frankly. I met someone named Valentino. And this is Valentino's story. It's an important thing from a perspective of a parent to know whether their child can see.
Valentino was born in 2006. He's 11 and a half years old. He was born perfectly healthy, normally developing, and then when he was about 3 and a half, he had a near drowning episode that he was diagnosed with a significant global developmental delay and significant traumatic brain injury. One of his many diagnoses was cortical visual impairment.
We didn't know whether he could see.
About a year after his incident, we started to look for options for treatment. We've met plenty of clinicians along the way who said, they look at Valentino in any one given, second, they'll make a decision. Oh, he can't do anything. This is just the way it is. This is the situation, accept it. So we started to look around in Hong Kong, where we live, around Asia, and also in New York, where I'm from.
Valentino's parents had heard of my work and asked to come see the research that happens at the Burke Medical Research Institute. His parents were most concerned about his inability to become visually aware, that he only occasionally followed their faces, When I took her to Glen Prusky's lab and she saw the method that he developed for testing rodents and their vision, she said, "My son can do that."
And what we found really helpful, and we're so grateful to have had this experience, is there was a group of people who understood the brain, who understood the intricacies of what we need to improve, and led us. The blind is not Valentino, it's actually us.
Valentino's parents were impressed that you could measure vision in rodents in the way that we did, and wondered if the technology or the approach we used was able to enable him to have his vision measured. Valentino's role is absolutely essential. This wouldn't exist without him, I think. Initially, he came and we kind of did a modified version of the rodent system for Valentino, where we had a camera that watched his eyes and we placed information on the screen and moved. And we were able to show that indeed he was able to track information on the screen and do it smoothly. And it was actually at that time the first time that we could really tell his parents that he could see. And over the course of the past 6 years, we first took it from the concept and a sort of a rodent-adapted system to a specifically human system.
The unique thing about working with this team is, I think it's just the tailorization of this concept. And one of the things that Valentino's functions actually that were left intact and actually quite sharp is his hearing. His penchant for music really motivates
The
him.
music really helps. Actually, he's probably very musical.
I took an ordinary computer monitor screen, as wide as we could find, and I used some of the software toolboxes that are out there for presenting very carefully controlled visual stimuli, and developed an algorithm that would take input from an eye tracking device and determine in real time whether the person's eye movements were smooth or not, and smoothed along the expected trajectory, and connected that to a system that played or paused a piece of music.
What these guys do, and I think this is very endemic to people who do research, scientists, is trying to find a better solution than what you have today.
The system, which we call optokinesis, is a device that enables us to measure visual function in children who otherwise vision measurements aren't possible in them because they're uncommunicative. So it's a new breakthrough, we hope, in our ability to measure children. And then over the years, Valentino has been regularly measured, and indeed we've shown that his visual function has vastly improved over that time.
It's part of our daily training for Valentino. For his vision. We believe and have seen that he's actually gotten better from this program, using this program on a very regular basis.
It's exciting. It's a sort of rather fast-moving facet of neuroscience.
From one child, Valentino, we've now tested about 30 children at Blythedale Children's Hospital.
Actually, by this measurement, we can say that not only can they see, but they can see as well as other children the same age.
The expertise at Blythedale, their care of kids with those kinds of disabilities, we're finding it a real delight. It's definitely far more advanced and reliable than the options that we find in Asia. Very truthfully, I'd love to bottle that up and bring it back with us. And yeah, Blythedale is kind of uniquely equipped to make a whole kid, better.
So what we want to do is to not only measure once, but measure and give experience many, many times as a form of therapy.
If modern medicine's kind of reached the edge of its ability to help, there are researchers who are dedicated to pushing that envelope.
There was nothing available, and now there is, and that's an incredibly satisfying achievement.
I would love for him to be able to meaningfully communicate, and that's something that we're also working with Blythedale and with Burke, and hopefully we'll get there.
Well, This truly stopped me in my tracks. Valentino's not here today, but Valentino's mom is actually here today from New York. I owe these people a tremendous amount of gratitude because it really got me into this business at all. And now most of my lab is working on this. I also, Blakedale Children's Hospital, we've collaborated with now going on 7 or 8 years. And again, I'm a Cornell professor, but it's this place and the wonderful place that it is that has enabled us to do this, not with only Valentino, but with many kids. And so basically what we've done, in a nutshell, is to take this rodent system and be inspired by that methodology to develop a new set of metrics and a new set of procedures to measure children with brain injury who are uncommunicative. So it's a very simple system. There's a computer monitor. We put information on a screen. We move that information. We track eye movements relative to that information. And there's a child. This is a child. Obviously, it's Christmas season at Blakedale Children's Hospital looking at this. I can go into a lot of details about why and how it's come to what it is, but you can see it's quite simple. And kids are looking at video monitors a lot in these situations anyway. The approach we take is we call it quantitative neuroophthalmology. And basically, I think of it like a ladder. That you've got, low functions, that could be like even pupillary responses and so forth. You got high functions, it could be, face recognition, color, fancy motion perceptions, and so forth. What we think is there's a way to use eye tracking technology to do different measures. I'm going to tell you today about these. There's those measures below there, though. We have a system that will detect pupils and test autonomic nervous system functions, and also working on ones above that. But today I'm just going to show you about visual fields, saccades, spatial vision, and so forth. And I'll do it very quickly here. We think eye tracking and fancy computer processing. We do this stuff in a frame-by-frame way using video card analyses that is happening in gaming all the time to do this. It allows us to do it quickly and so forth. Here's sort of an idea of what this visual ladder does. They're stripped down little versions of what we're doing. The green marker will show you where the gaze is, but that isn't on the screen when we do it, but just show you how gaze can control this. You can see as you move your eyes around, you can pop these bubbles. It's a simple little game. Here's a thing called bubble burst. When you put your gaze on it, it'll pop those bubbles. That elicits saccades, and we do this until we get enough saccades to measure it. We do the same thing with tracking, so we can detect smooth tracking and quantify that. Visual field, we know where you're looking because of the gaze. We can put things in different visual fields, so we can test whether you got holes in your visual field. Here's a thing called gradiate, where you have information streaming on a screen, and we measure acuity or contrast sensitivity from that. And another one here where you can use balls to do the same sort of thing. I'm not going to go into the details in this, but this is the ilk of what we're doing. And from this, we can do a number of quantitative analyses about not just where you're looking, but how you look, and how your look or gaze changes as information on the screen changes. So for example, these are saccades. These are the fastest movements our bodies can make, and we use them to move around visual world to put our fovea in that information. And the typical pattern that we have for normal individuals is like this, where you have a bow tie kind of thing. You move better on the horizon. It's also a rectangular screen. And so you can see you have more movements and so forth. The other thing is, I'm showing you on each one of these slides children that are communicative and non-communicative. So you can see here is a typical bow tie kind of configuration, where we move better on the horizon than up and down, and quantify it in a variety of orientations. Here's unusually isotropic. They don't have that typical horizontally biased set of saccades. Here's a situation where there's distortion from head tilt that we're working on ways of correcting. And here's pathological nystagmus. Nystagmus, people have seen and heard about that, right? That's a very common thing. Well, we can now not only say you have nystagmus, I'll show you in a second, but how and which direction, how quantitatively changed it is. We can calculate and analyze the saccades not just by where you're looking, but the distance over which you look, and the same sort of thing. That you can have the normal kind of patterns that you see, and you can have atypical patterns, and those atypical patterns are quantifiable. So we set out a set of metrics that are not only quantifiable, but that a non-expert can look at quickly and see where there's a change. That's very evident here in when you look at, say, the biases. So if you have saccades, you can see that there is a bias in the leftward direction. Because many brain injuries occur in a lateralized way, you often have asymmetries. This allows us to not only say there's an asymmetry, but quantify how much that asymmetry is, and you can have the opposite and so forth. The same is true if we don't move the information. When you look at this moving bubble, these pursuits, you see that you can have biases in certain directions with suspected intrusion of nystagmus, for example. So if you don't move the bubbles, like remember the moving bubble one, you have to track it for a while, but if you just put the bubbles on, but let's say someone has a standing nystagmus, you can see that. And again, here we've calculated not just that you move in atypically in a particular direction, but the arrow tells you which direction and its length tells you how much it's different. So now we have a way of measuring not once, but repeatedly, and even in circumstances where you have treatment at hand. The moving bubbles thing is a much more difficult thing to quantify, but normally what you have is a system, again, of these different orientations and degrees of movement in those orientations that we can quantify. The field bubbles, looking at your ability to perceive things in different visual fields, we can calculate not just spatially in visual fields, but how quickly you move in that. And so here is an array, and you can see that white means good, basically, and the more penetrant the color toward red is, tells you how much not good. And you can see in the lower and right-hand sides, there can be complete holes in that world. And you can see that even in individuals that have can see in all orientations, they don't see as well. They aren't able to orient as well to that information. Again, a quantification of that. So you can see that there's mild impairments in some children, and there's major ones in others. In fact, this child right here, the C6, is legally blind. And you go, well, how can they see? They're just moving their eyes around the screen and all over, right? They're doing all parts of the screen, but they're not doing it, we think, based on what's on the screen. I'll show you that in a second. And these, of course, are highly impaired. Here's the same situation, except now we look at how directly you move to a dot on the screen. And you can see that this child down here, the C6, now is just very impaired because they aren't moving directly in relation to that stimulus that's there. This function, I said, acuity and contrast sensitivity. We love contrast sensitivity. I go on and on about contrast sensitivity. It's not measured regularly, it should, and the reason it isn't is because it's very difficult and time-consuming to measure. So we've come up with a very simple, quick way of doing it, and now we can measure these very complicated things, even in children, as you can see here, that are highly impaired and impaired enough that are uncommunicative. And so these two top rows here just show that whole field one with those kind of dots moving versus the one on top, which is the ball movement one. But basically you end up with these arcs, and the distance on the arc and how far away from the zero point in the corner there you are tells you how well you see at different scales. And so we can measure these highly difficult to measure things in children. Here is our superstar for this. We've got about 4 children now that we followed for a great time. We've measured about 200 children in total. And you can see this child came in. This is a gunshot wound. Child survived. They thought they were going to pass. They actually removed life support. The child somehow lived. Came to Blythedale about 3 weeks after this. They were uncommunicative for 2 months, but still there was enough ability to do these tasks, this fairly complicated task, if you will. And it just kind of pulls your eye over there when you see movement, and we only need a fraction of a second to get information about how well they do that. And you can see from the outset, even when they're uncommunicative, you can see that they're able to be measured on the full screen or the ball version of this task. And look at the improvement over time. This child walked out not under their own power, but with some help, 9 months later after this. And look at how much better they can see than when they came in. I can't claim that our efforts enabled them to see better, but we followed their ability to see over time and in a quantitative way. This is exactly what we're going to. So, finally, I hope I've convinced you that we've got a long ways to go, but we can now compare humans to animals. We have to do that because we use animals to model the things, and if we're measuring them in different ways, now we can measure them using the same kind of approach. So we hope that this will improve our translational ability. It's been a woeful problem to translate things in animal models to humans. We hope that if we're measuring apples to apples across the species boundaries, it'll be better. We also think young to old. So we have to measure the development of these things for health span studies before we can differentiate normal development from disease states, of course. Communicative to non-communicative. Again, the heads are nodding because this is a huge problem. We hope it's not a panacea. There's some children you simply cannot measure, but we've expanded the ability to measure children and take that boundary out. And so comorbidities, CVI always comes with other things, right? And so we hope this will allow us to separate those things, and we hope to be able to define CVI actually better with much larger case studies or clinical studies that we've done. Healthy disease, so we can sharper define these things and do it quantitatively, and where we really want to go is treatment to control. So we need to quantitatively evaluate these therapies that we're wanting to test, and that's how we'll move forward in this field. So I acknowledge I have a wonderful lab filled with fantastic clinicians, computer scientists, computer engineers, and I've been very grateful not only to the Sandras and those parents who have enabled us to do this work, but the National Institute of Health through the NEI particularly, we have received wonderful funding and support from them for this. And of course my institutions, the Burke where I work, Cornell Medicine where I'm appointed as a professor, and at Blythedale. And I thank you very much for your attention.